Good afternoon, everyone, and thank you for being so punctual. And thank you, Chandra, for starting the recording. I guess we can just begin, and hopefully, if there's others that trickle in, they won't be too disruptive. Thank you for joining us today at this National Biodiversity Assessment webinar 2025. It's the sixth webinar of the series focusing specifically on the genetic component of the MBA. Myself and Alex will be your speakers. I'm a principal scientist within the Zoological Research Unit at SANBI, where I basically use molecular tools to help inform wildlife management. I'm also part of various international consortia, including the Coalition for Conservation Genetics, GeoBond's Genetic Composition Working Group, and the Conservation Specialist Group of the IUCN. And all of these are actively involved in developing and testing some of the genetic diversity indicators now being used for biodiversity reporting and monitoring at both national and global levels. And since NBA 2018, I took over as the lead of the genetic component of the NBA and have coordinated the inclusion of some broad genetic initiatives South Africa is involved in, including wildlife forensics, DNA barcoding, and biobanking. So I encourage you to look at those on the NBA website. And I've also coordinated the genetic indicators that we'll be presenting here today. And I'm going to hand over to my colleague, Alex, for an introduction.
Hi, everyone. Yes, thanks, Jess. I'm Alex Nieto. I'm an applied research officer at Anura Africa, which is an amphibian-focused conservation NGO. And I'll be discussing some of the contributions that I made to the latest NBA from my master's dissertation from last year, which I completed through the University of Johannesburg and SANBI. But yeah, Jess is going to set the stage for us now before we dive into the actual indicators. Thanks.
Thanks, Alex. So these webinars are but one product produced from the NBA. The main output is a book that you can download. And for the very first time for this NBA, a website with 108 web pages and thousands of images and plots and maps. Regular updates of the NBA via this website are now possible by the NBA team themselves. So the data science skills we have built over the last 4 years have allowed us to take a huge step towards a living national biodiversity assessment, which is very exciting. And we will use this momentum to improve our biodiversity monitoring systems in general. All of this being at the forefront of biodiversity assessment requires constant innovation and problem solving with few global examples for us to follow. It also requires collaboration both in South Africa and with international scientists and institutions. Oops, sorry. This brings us to a really big key aspect of the NBA. It's clearly an enormous undertaking. NBA 2025 was a collaborative effort from over 490 contributors representing approximately 110 institutions. It took approximately 23,000 person days of work over 6 years. And so it requires a great deal of facilitation from the leadership team and quite amazing goodwill and interest of our partners in making this a useful product and process. So I really do thank everyone who has contributed in any way to the NBA. And this is a product of the whole biodiversity of South Africa. So I really hope people will really acknowledge that. Okay, so now we're going to delve right into things. When we think about biodiversity, we often focus on ecosystems and species. But biodiversity actually has three interconnected pillars. We have ecosystems, species, and genetic diversity. And genetic diversity is the variation within and among species and its populations that allows organisms to adapt to changing environments. It also allows them to resist disease and basically persist over time. Without genetic diversity, species may remain present in the short term, but could lose long-term resilience and adaptive potential. So importantly, protecting ecosystems and species does not necessarily ensure that their genetic diversity is protected. Populations may become fragmented, isolated, or genetically depleted long before species are considered threatened or ecosystems appear degraded. So biodiversity cannot be fully understood, monitored, or conserved by focusing on on any one of these pillars alone. All three pillars are essential. They're interconnected and complementary components of biodiversity conservation. However, until recently, genetics has been underrepresented in South Africa's National Biodiversity Assessments. We have been working to address this gap through the integration of genetic indicators. While reporting on the genetic status and health of South Africa's biodiversity began in the previous NBA 2018, this work expanded substantially during the current assessment cycle, driven in large part by the adoption of genetic diversity indicators within the Kunming-Montreal Global Biodiversity Framework. And it's a process which SANBI and South Africa more broadly really played an active role. And NBA 2025 therefore represents one of the first attempts to operationalize multiple genetic indicators at a national scale. So in today's webinar, we will explore what these indicators are, what they tell us, and why they matter for understanding and conserving biodiversity. And today's webinar is structured around four genetic indicators currently included in the NBA. They operate at different biological levels and answer different conservation questions. Alex will begin by taking us through two higher level genetic indicators that focus on evolutionary potential and patterns among species. And I will then take you through two population genetic indicators that focus on genetic diversity, within species genetic diversity, connectivity, and long-term population viability. Together, these indicators help provide a more complete picture of biodiversity and genetic resilience, as no single indicator captures all aspects of genetic diversity, just like none of the three levels of biodiversity capture the full suite of them. So I'll hand over to Alex.
Yeah, thanks. Discussing these two indicators, edge and expected loss of phylogenetic diversity. This is the first time we're reporting these metrics in the NBA, so I'm going to dive into a bit of a background about them before showing the findings. So phylogenetics is the study of evolutionary history, and the first question that might come to mind might be, what is the conservation relevance of evolutionary history? Why should we care about this unseen information apart from what it tells us about what a species is and what it's not? And the key reason for this is that different species hold different evolutionary histories that are both unique and quantifiably unique. Not all species are quantifiably unique. And this can be attributed to differences in mutation rates or perhaps species with many closely related sister taxa. To put this into a conservation example, if you had 4 species here that are closely related, 3 are least concerned, one is critically endangered, and then one more distantly related species that's also critically endangered. If you only had the conservation capacity to preserve one of these species, you'd say, okay, it's not that or unique one. But if that species were actually vulnerable, can you in good conscience put all of your capacity into protecting this species, knowing the other one will certainly go extinct? But if it did go extinct, it may be functionally replaced by its sister species. And so the challenge becomes, how do we quantify biodiversity in a way that captures not just the number of species and which ones are threatened, but how much irreplaceable history they represent and how much is at risk? And this is especially important given our limited capacity to project everything. The original papers discussing it called the agony of choice. So how do you quantify this? For a single species, PD is essentially the sum of all branch lengths connecting it through evolutionary time. The branches on these trees can represent genetic differences, but they can also be time calibrated to represent millions of years. So PD is quite literally giving an indication of how many millions of years of a species represents. For a group of species, their phylogenetic diversity is some of all things. There are a few benefits to thinking about conservation from this angle. Some studies have shown that declines in phylogenetic diversity have been linked to cascading effects on ecosystems and on nutrient cycling. Another is that it is nearly entirely free of taxonomic bias. So for species, groups that have questionable taxonomy, where experts can't agree with one species should represent 10 or one different species, PD takes the question out of it and simply asks, how much does this organism represent? And then linked to this benefit is that by preserving groups of species that haven't been fully taxonomically resolved, we're also preserving the potential future values for humanity. Although this may be more relevant to plants that have emerging medicinal applications. PD, however, does not account for humans. A species with many close relatives does not represent the same level of irreplaceable history as a species sitting alone on a long isolated branch. So the natural next step to this is evolutionary distinctiveness, which uses a fair proportion approach to weight branches according to how many descendant species share, bringing attention now to uniqueness and irreplaceability rather than absolute diversity. A lot of other metrics have been designed based off of these two concepts, many of which I covered in the dissertation. But the way these two metrics become actionable priority indicators is by incorporating extinction risk data from the IUCN Red List. By multiplying risk into our component for phylogenetic diversity, we can get an indication of how many branches of the phylogeny, or how much evolutionary history, is expected to be lost in the next 50 years. On the other side, using evolutionary distinctiveness at its base, the evolutionarily distinct inverted engagement method is most commonly used to identify priority species that are both genetically irreplaceable and in urgent need of conservation. More recently, it's been expanded from this species priority mechanism to an indicator that assesses, or that can assess the state of all edge species for a particular group or for a nation. Both of these are adopted in the Global Biodiversity Framework, making them incredibly valuable for reporting on our contribution to global conservation. Furthermore, South Africa is in the unique position where we have reassessed species periodically for different taxon groups. So we can now monitor how these indicators have changed over time to see conservation may have an impact on preserving novelty. I am hearing that my audio is a bit inconsistent. I will just try quickly switching mics and then feedback and stuff like that. Okay. And you can?
Just try it out and we'll see if it's okay.
Okay, sure. I'll keep going.
Sounds good.
All right. And so, yeah, because we can now track this, we can see if conservation has been having an impact on preserving genetic novelty, or if threats to extinction have been increasing across assessment periods, similar to how the Red List Index operates. So jumping into the first indicator, the expected loss of phylogenetic diversity can be expressed in a few different ways. The reportable index is expressed as the percentage of the total phylogenetic diversity of the group expected to be lost. Using South Africa's amphibians and reptiles as a case study, we can see how if no conservation interventions are implemented, South Africa stands to lose approximately 5% of reptile phylogenetic diversity and 6.5% of amphibian phylogenetic diversity within 50 years. This may not sound like a lot, but this 5%, for example, represents 689 million years of permanent loss and loss of evolutionary potential, information, and functional traits. As you may have also noticed, the trend for this expected loss is near stagnant, which can potentially point to a few different things. The threats to species may have already been well established by the point of the first assessment and have not been reduced. This does align with our land use data, having most of the land use change occurring prior to 1990, but localized threats are definitely emerging. The other option is that there could be a lack of threat intervention or successful threat intervention, or perhaps species are still in extinction depth, where the consequences of threats emerging 50 years ago may only be felt in the decades to come. Overall, it is clear that there is a need for strengthening targeted conservation efforts for South Africa's threatened fauna diversity. But what is this diversity and where is the target? What are we supposed to be protecting? And this is the component that is being piloted in South Africa. That is to map this predicted loss spatially, identify priority regions for conservation. So this was done for both amphibians and for reptiles, mapping concentrations of phylogenetic diversity that has now been weighted by probabilities of extinction. And this allows us to visualize where phylogenetic diversity is most at risk. For amphibians, the highest or high concentrations can be seen in the temperate southwestern Cape, particularly among these fragmented mountain habitats. You can even see a shadow of the Cape Fold Belt coming across here. More prominently, however, is the risk shown for the subtropical northeast of the country, where there is a much higher concentration of species, as well as a large number of threatened species, resulting in the high risk that we see here. Interestingly, reptiles show similar trends, where the highest concentrations are also found in the more subtropical northeast, but now particularly in Limpopo, on the South Hansberg Mountains, and on the borders of Mozambique. Some cases, like here, can be separated from the species that is causing it. In this case, it's Cryptoblepharus africanus. This species is actually a unique case where its full range extends further into Mozambique. And if we were assessing it, this indicator or the species within this broader context, the species would be least concern and we would completely dismiss this function. But because these are national indicators and because its distribution within South Africa is so restricted and the land is so threatened, it is assessed as nationally critically endangered and the region is appropriately emphasized here. Moving over to the next indicator, the EDGE species concept has been a priority tool since 2007, but few countries have been able to produce comprehensive national lists of EDGE species for any taxonomy. The main reason being a shortage of data. To generate this list, you need a comprehensive national phylogeny, which in turn requires genetic data for all species in country. And then nearly all species need to be assessed for extinction risk. This is a tall ask, so we are among the first countries to conduct the national assessment like this. Here are two shortened lists for the top EDGE amphibians and reptiles of South Africa. Although the risk of extinction is weighed heavily in the calculation of an EDGE score, the reptile list demonstrates how even a comparatively less threatened species can be a more pressing conservation target as it represents more genetic. Similarly, for the amphibians, you can see here the top 5 EDGE species are all endangered. And that's not to say there aren't plenty of critically endangered species that need protecting, but these have been selected based on the irreplaceable loss that they would represent. But if we were to map these, looking at the density of EDGE species across the country, we can see that reptile EDGE species are generally quite spatially diffuse, requiring distinct targeted conservation. But for amphibians, we see as many as 5 EDGE species concentrated here on the Cape Peninsula in one grid cell. The CBD reportable component of EDGE is known as the EDGE Index. A workshop in collaboration with the French Agency for Biodiversity was held in 2024, where we discussed how components such as the EDGE Index should be calculated on global versus national scales, depending on the availability of genetic data and extinction risk data. South Africa's representatives were strongly involved in this discussion as we had the data that would form one of the primary case studies for this new methodology. How the EDGE index is essentially calculated is the proportion of genetically unique species that are threatened out of the total number of distinct species. So with that in mind, the case study that we generated from this was a comparison between what the EDGE index might look like if you used globally disaggregated extinction risk. That's red list data that is globally calculated. Or as in the case within South Africa, you use these nationally generated red list data. Amazingly, we had the data to look at this for all of South Africa's vertebrates. And what it showed was that EDGE indices are generally higher when based on nationally produced red lists. These are the nationally produced ones here. Additionally, the confidence intervals are generally smaller for the national data sets, and this disparity is due to a large number of species being categorized as data deficient or not evaluated in the global assessments, whereas on the national level, we have actually assessed species. So together, these higher EDGE indices and lower confidence intervals suggest the national data sets are more sensitive to actual threat status and are more reliable in producing the most accurate EDGE index for a group. We also assess this for only the endemic portion of the species to see how the EDGE index might look. So I've placed some markers here so you can see how it changes when we look at only the endemics. And you can see how for nearly every EDGE index, there has been the EDGE scores get higher, indicating that evolutionarily distinct endemics are proportionally more threatened. Similar to the expected loss, PD loss indicator, because we have multiple assessment periods for amphibians and reptiles, we can map the trend of the EDGE index over the last 20 to 30 years. In this case, we observe a similar thing. There has been very little change over this period for both grades, suggesting a lack of successful intervention efforts and an urgent need to strengthen targeted conservation. Looking at the actual values here, where the darker colors represent the entire group and the lighter ones are the endemic portion, we see in the same case as the vertebrates, that the endemics are disproportionately threatened when compared with the broader groups. For example, the amphibian endemics, 41% of the endemic evolutionarily distinct amphibians are threatened with extinction. So that is a broad overview of what these above species level genetic indicators look like. Now Jess is going to jump into what's the below species level genetic indicators look like and how we reported that in the NBA. Thank you very much for listening.
Thanks so much, Alex. We'll now move on to the genetic indicators below the species level. They are the populations maintained, or NE 500. These are the shortened terms to explain these indicators. In full, they're actually the proportion of populations maintained within species and the proportion of populations with an effective size, or NE, above 500. So we'll first go into each indicator to provide some background on it. The populations maintained indicator basically looks at whether populations historically present are still maintained. So it's asking, is there population loss? Populations that are geographically or ecologically isolated often develop local adaptations and unique allele frequencies. So in this image to the left, this is indicated by the different colors of trees. So maintaining distinctness protects these adaptations. But as populations are lost, their ability to withstand changing environments is likely to decrease. So we want to maintain as many naturally distinct populations as possible. This is in contrast, of course, to anthropogenically isolated populations, which can create artificial genetic structure or distinctness, which is not what we want. And an important point here is that species can remain present while still losing substantial genetic diversity and structure. So in other words, a species may still exist on the landscape and may not yet be considered threatened based on demography, but populations within that species may already have disappeared. So this can reduce the species' overall adaptive potential and resilience long before the species extinction actually occurs. And so while the populations maintained indicator looks at how many populations exist, the NE 500 indicator looks at whether the proportion that remain are large enough to retain genetic diversity over time. So an effective population size is the number of individuals that essentially contribute to the gene pool. So the ones that actually reproduce, so to speak. And so an NE of approximately 500 individuals is widely considered within the population genetics and conservation genetics communities as being an important benchmark for maintaining long-term adaptive potential and limiting the effects of genetic drift and other factors. So populations above this threshold are generally considered more resilient over the long term, while populations below will tend to face greater genetic risk. Importantly, NE 500 is not a hard extinction threshold. Rather, it is best viewed as a monitoring benchmark and an approximation of the population's viability. One reason this indicator is particularly important, as I've already mentioned, is that demographic recovery does not always mean genetic recovery. A population may increase numerically in numbers, but it may still remain genetically poor. It could be because of it being highly inbred, which could also be because it started from a small number of breeding individuals, essentially a small founder population. And so this would cause its effective population size to remain low. So in a sense, NE can sometimes provide a more realistic picture of the genetic health of a population than census size alone. Both of these population level indicators benefit most from the use of actual genetic evidence. So this is the gold standard. Genetic data provide the most robust and biologically informative assessment of population structure, connectivity, and genetic health. However, for many species, genetic data sets are still limited or completely unavailable. And this is not just a South African problem, but it's a global one. This does not impede these genetic indicators from being calculated, which is a benefit of these indicators. Demographic, ecological, or biogeographic data can be used as proxies for genetic information. For example, information on the distribution patterns of a species, the biogeographic barriers, the dispersal ability can all provide insight into the likely population genetic structure within a species. While census size or density estimates in capture mark recapture data can be used to quantify effective population size in most instances, I'll go into that in a bit. But essentially, these alternative data sources are not importantly considered equivalent to genetic data, but they do allow these indicators to be operationalized more broadly across taxa until such time whereas genetic data can be acquired. There is one important caveat, especially for the NE500 indicator. When species are known to have undergone a bottleneck, so they've gone through a population crash, they generally experience a severe loss in genetic diversity, not only in their population numbers. And once they start recovering, the census information could be misleading, given that, as I mentioned previously, populations may increase, but their genetic diversity hasn't had time to catch up. So therefore, only genetic evidence could be used in such instances. So when there's a major collapse, and then really just trying to track that recovery. So these indicators would not be that effective. Okay, so now going back to how this indicator, specifically the effective population size indicator, could be estimated without genetic data. This can be approximated from census size, as I mentioned, using a conversion ratio. A commonly used default ratio value is 0.1, meaning that the effective population size is estimated to be roughly 10% of the number of mature individuals that exist within a population. So not the species, but in a specific population. And under this scenario, the effective population size threshold of 500 would correspond to approximately 5,000 mature individuals. Importantly, this relationship could vary among species and taxonomic groups depending on various factors such as mating systems, reproductive output, generation length, and other life history traits. Because of this uncertainty, it is best to evaluate multiple conversion ratios just to provide confidence in your reporting. So we would generally do the default 0.1, but in most cases it would also range from 0.2 to 0.3, and these range from more conservative to more liberal estimates. But encouragingly, the overall national value tends to remain relatively robust. It doesn't really change except for maybe 0.01 difference most times. So that provides the confidence that the indicator still holds broad trends, captures broad trends in long-term genetic resilience. Okay, so now that I've given you that background on these specific indicators, you're ready to see how South Africa is faring. The values posted in the recent National Biodiversity Assessment are based on a pilot study South Africa featured in to develop and test these indicators. It was based on 126 taxa across 8 different taxonomic groups. However, data wasn't necessarily available for all the taxa that were assessed on purpose. And so the values I'm just going to layers pointer. The values you see here reflect the actual number of species that contributed to those scores. The indicators show that the vast majority of populations are generally maintained, which is really great, but the majority have populations that are too small to maintain genetic diversity, which is concerning. Before really moving on, it's important to acknowledge that these indicators, while being informative, are still developing and come with some data gaps. So one of the biggest challenges, as I already noted, is that many species still lack genetic studies altogether. So even fairly charismatic groups, such as mammals and birds, have fairly limited information, at least based on what's published. As a result, the mass majority of those previous assessments are still based on demographic and ecological proxy data, but even then, available data was limited where there was a few. And so I already pointed out with the sample sizes. However, the indicators allow for this uncertainty. So it's a limitation, but also a benefit because they're able to factor that in. Another major challenge for anyone working with these indicators is defining what a population is and what their boundaries are in a biologically meaningful ways, particularly for highly mobile or poorly studied taxa. And this is not necessarily unique, given that even species designations can often be contested, but there's a growing body of work that is helping to address this. An additional important consideration apologies, is that transboundary populations were not incorporated into that first assessment. So this likely contributed to the relatively low NE values that we observed, NE 500 values that we observed, because many species naturally occur across political borders and may form part of a larger connected population. So these limitations do not by any means mean that the indicators are not useful. Rather, they simply highlight where future investment and development are needed, and expanding genetic sampling, improving long-term monitoring, strengthening data mobilization, and really integrating genetics more fully into biodiversity assessment workflows will all help improve the robustness and coverage of these indicators over time. A number of important developments are now helping address the vast majority of those challenges and really improve the operationalization, there we go, of genetic indicators nationally. One major development has been expanding the taxonomic coverage beyond isolated case studies. For a megadiverse country such as South Africa, representation even from 126 taxa was quite limited. So we really strived to address this. We have now completed the indicator process for South African mammals. Amphibians are currently underway, and additional taxonomic groups will progressively follow. A second major development has been the use of existing conservation assessment processes, particularly the red listing, and as a mechanism for large-scale data mobilization. So many of you will already be familiar. The Red List brings together experts. It brings together the most up-to-date published and unpublished information on species. It's just the most up-to-date distribution records, pretty much everything you can compile within a structured assessment framework. And importantly, it also helps integrate genetics directly into mainstream biodiversity assessment workflows, rather than keeping genetics isolated within individual research projects. And a third important development has been improving the biological realism of these indicators by incorporating transboundary populations. So together, these three different developments are helping strengthen the robustness, scalability, and practical utility of indicators, which we hope to inform management, but also provide more realistic updates on the MBA website. And just to give you a quick example, South African mammals, as I mentioned, is just completed. And this is one of the first examples globally of implementing these two indicators across an entire taxonomic group within a country. So in total, this assessment included 320 assessments, covering 293 species and 27 subspecies. Using the populations maintained indicator, we found that approximately 98% of historically recognized mammal populations are still existing. And for the NE 500 indicator, approximately 82% of assessed populations were estimated to exceed the NE 500 threshold once transboundary populations were incorporated. So importantly, incorporating those transboundary populations had a major influence on the results. Earlier pilot analyses did not account for those, and it estimated that for mammals it was 61%. But just to point out, when the transboundary nature of those same assessments of these assessments, the whole taxonomic group assessments was removed, it went down to 66. So 61 to 66 isn't a major difference. So that initial pilot method still showing that the methods are quite robust, but incorporating the transboundary nature really does provide a more realistic view of what their genetic health might be like. Another important finding was that signals of genetic erosion were not strictly only to highly threatened taxa. While critically endangered and endangered mammals generally showed the greatest genetic risk, which is not surprising, some vulnerable, near-threatened, and even least concerned taxa also showed evidence of long-term, so these groups over here also showed evidence of reduced long-term genetic resilience. This highlights an important point that traditional threat assessments and genetic indicators provide complementary information. Species may appear relatively secure demographically while still experiencing erosion of genetic diversity. So a similar plot to this one exists in the on the NBA website, but just for those reduced data set, but it shows a very similar picture. And overall, these results demonstrate that national scale genetic assessments are feasible, informative, and scalable, and they provide meaningful insights that can support biodiversity monitoring, conservation planning, and evidence-based decision-making, both nationally and internationally. Okay, so that's essentially the end of our main presentations. We just thought we would leave you with a few key messages. First, there's no single indicator that captures all dimensions of biodiversity. Biodiversity exists across multiple interconnected levels, including ecosystems, species, and genetic diversity, and these dimensions are complementary rather than interchangeable. Second, different genetic indicators provide different insights. So higher level indicators such as age and expected loss of phylogenetic diversity helps us understand the importance of the evolutionary history across species and the unique history within South Africa and how the loss of any of it would impact the country. While population level indicators such as the populations maintained indicator and NE500 really provide insight into the genetic health and long-term resilience within species. So together, these indicators provide a more complete picture to our biodiversity status and change. And then there's a third take-home message is that genetic indicators can reveal hidden dimensions of biodiversity risk that may not be detected through the more traditional conservation assessments alone. Genetic erosion can occur long before species are extinct, and even taxa that appear relatively secure may still show reduced long-term genetic resilience. And finally, genetic monitoring is increasingly becoming operational at a national scale. Through improved data mobilization, increased investment in genetic data, and expanding our taxonomic coverage, these indicators are rapidly transitioning from research concepts into practical tools that can support biodiversity monitoring, conservation planning, and decision-making. And so ultimately, genetic diversity is no longer the invisible component of biodiversity conservation. It's becoming an increasingly important part of how we understand, monitor, and conserve biodiversity in the future.
Yeah, thanks, Jess. The MBA and many of its components would be possible without the contributions of many different individuals with vastly different skill sets and backgrounds. And this is no different for the genetic component. There are many opportunities for involvement and collaboration moving forward, particularly around filling the research gaps, as Jess mentioned. Species still lacking genetic and ecological data needed to evaluate these indicators. For students and early career researchers interested in this type of work, there are opportunities to help expand species coverage and improve these data sets through internships, postdocs, PhDs, and postdoctoral collaborations, particularly in the fields of biogeography and genetic diversity monitoring. There are also opportunities for data sharing, joint analyses, co-supervision, and workflow development. If you are interested in forming any collaborations, whether it's for contributing data or capacity, please feel free to reach out to Jessica. Finally, these indicators are not intended to just be academic exercises. A major focus now is supporting their application in biodiversity monitoring, red listing, conservation planning, restoration, and protected area management. So if you're interested in contributing, we would love to continue these discussions and explore opportunities for involvement. Thanks everyone so much for listening to this webinar. So we now have a few minutes to answer any questions or inputs from you. And while you're thinking about that and putting your hand up, I'll end on a quote by the Deputy Minister at the launch event of the MBA. The National Biodiversity Assessment gives us the knowledge to act. Now it is up to all of us to turn this knowledge into lasting change for nature and our people. Great. Thanks everyone. Happy to take questions.
Thank you, everyone. Does anyone have a question that comes to mind based on our presentations today? If not immediately, you can also mull it over and then contact us afterward. Okay. I did try to look in the chat, but I couldn't see anything. So yeah. Great. Alex, do you want to close us off?
Sure. Yeah. thanks everyone for attending. Please note that all the recordings of the webinars will be uploaded to Sandy's YouTube channel, where you can share them. We encourage you all to delve deeper into the MBA, and please reach out if you want to participate. Thanks, everyone.
Thank
That's all.
you, everyone.